Prions are scary. My name is Colin, and my group members, Charlene, Meyer, Kayla, and I will be presenting on prions and prion diseases. First, I just want to give you guys a little background on what prions are. Basically, prions are the infectious agent for prion diseases. These diseases include Creutzfeldt-Jakob disease, or CJD, scrapie, mad cow's disease, and kuru. As you guys may know, the infectious agent for viral diseases are things like DNA or RNA, and the infectious agent for bacterial diseases are bacteria. Prion diseases have a different infectious agent, and I'll give you guys a couple seconds to guess which one. So it's not DNA, it's not RNA. The infectious agent is misfolded proteins. The protein itself has two main conformations. There's a normal protein, PRPC, and the misfolded protein, PRPSC, which is the disease-causing conformation of the normal protein. You might be wondering how you get from the normal to the disease-causing version of the PRP protein. This can happen a few different ways, the first being through mutations in the PRNP gene. The PRNP gene is the gene that encodes for the PRP protein, so mutations in this gene can predispose the normal prion protein to misfolding, which results in the PRPSC. There is also sporadic misfolding, where a normal protein changes due to random errors in protein folding during cellular processes. Self-propagation of prion proteins can also happen where a misfolded protein induces changes in a normal protein. Lastly, you can be infected by the misfolded protein by eating raw or undercooked food where the animal you are eating had prion disease. This could be beef from a cow with mad cow disease, which is a type of prion disease. Prions in the past. Stanley B. Prusiner was the main person involved with prions in the past. It started with confusion about CJD and scrapie in humans, because these diseases did not fit the normal models of infectious diseases at the time, which had infectious agents like DNA, RNA, and bacteria. Dr. Prusiner noticed that the TSE was highly resistant to heat and radiation, which led them to believe that these diseases were not caused by typical viruses or bacteria. In 1982, Dr. Prusiner proposed the prion hypothesis, and he also named the infectious agent of prion disease prions. The prion hypothesis was the concept that diseases can be caused by infectious proteins alone, and it had not been previously proposed. This caused a lot of skepticism in the field. Over time, more evidence was discovered that supported the historical prion hypothesis, and in 1997, Prusiner received the Nobel Prize in Physiology or Medicine for his work. The current prion perspective is just a more supported version of the past prion hypothesis due to advancements in the fields of prion research.
Now let's talk about the different ways that prions can be transmitted. First, you can get prions from eating them. You can also get prions from a contaminated blood transfusion. A doctor might accidentally infect you by using contaminated equipment. There are also some mutations that increase the risk of your proteins misfolding into prions. And finally, if you're really unlucky, your perfectly normal brain might pull an oopsie and fold a protein incorrectly, and guess what? Now you have prions in your body. Regardless of how prions appear in your body, they'll eventually make their way to your brain. Once the prions arrive at the brain, this is where the interesting things start happening. Here they'll meet the protein we've previously defined as PRPC, cellular prion protein. As we've mentioned before, the prions themselves are actually a misfolded form of PRPC, called PRPSC. This basically means that they're pretty much the same protein, just folded into different shapes. We tried to depict this in our drawings, but PRPC is more rich in alpha helix structures, while PRPSC is more rich in beta sheets. And when the proteins have different structures, they'll also have different characteristics. Unlike PRPC, PRPSC is not soluble in water, it is resistant to heat, and it can even resist getting destroyed by enzymes such as proteinase K. The following two unique properties of prions make them especially dangerous and capable of causing disease. Number 1, they're able to cause normal prion proteins to misfold into more prions. And number 2, they can aggregate. That's right, prions will clump together to form aggregates of various shapes and sizes. Interestingly, recent research has shown that the structure of these aggregates is actually linked to the kinds of disease symptoms that they cause. A team of researchers was able to separate out some of these different kinds of prion aggregates, and they put them in some mice to basically give these poor rodents prion disease. And they found that the disease symptoms that the mice had were actually really different from each other depending on what kind of aggregate that they got infected with. And these ranged from hyperexcitability down to lethargy to kind of none of that. And when these ill-fated mice finally died, the researchers took their brains back out and they found that these different kinds of aggregates, they even found different hiding places in the brain. So clearly, prions can cause a lot of trouble by aggregating, but how does this aggregation actually happen? Well, another team of researchers has got our backs. They used some really cool technology called cryo-electron microscopy to actually take a look up close at what's actually going on inside these aggregates. In order to form those aggregates, prions need some way to stick to each other and clump together. And when the researchers took that deep look into the structure of the aggregates, they found at the very center something called hydrophobic interactions. These are essentially interactions between the prions that hold them together. So that would explain how the aggregates hold themselves together. But how do they get more prions to join them? Well, it turns out these hydrophobic interactions aren't just happening at the center of the aggregate. There are also spots along the outside that are also hydrophobic, and these will do these interactions with free-floating prions and drag them into the aggregate, building itself up. And as these aggregates keep growing, it is not kind to neurons. Normal PRPC is produced in the soma, up in that big cell body up there, and it gets sent down the axon and mailed out of the axon terminals to serve as a signal. However, we know prions love to aggregate, and aggregate they will right there in the axon. They'll start swelling it up and blocking off all of the functions, and well, that's not good for the neuron. Unfortunately, the neuron will not be surviving this disturbance.
No, no cure for prion diseases, so you're kind of stuck with them if you have them. There is treatment though, not to get rid of them, but to make you feel better, just for your symptoms, you know. So, main one is mental health care because some individuals may be feeling depressed, some anxiety. So, taking care of their mental health, whether it's counseling, support groups, or medication, just making sure that they feel comfortable because these diseases are a lot to deal with. Then you have pain management because this is a neurological disease, can have muscle spasms, headaches, mood behavior change, um, different types of like stiffness or coordination issues, sometimes epilepsy also. Um, so that's just like medication management and different types of therapies to help individuals cope with those things, and just overall supportive and palliative care because these diseases are super fast progressing and they're also very, very fatal. A lot of individuals do not live for over a year. Most die within like 6 months. So, just improving their quality of life while they're still alive and giving the best end of life care is basically the whole entire goal of treatment. So, some upcoming and future treatments. Kind of cool, right? Some gene therapy. Trying to use gene therapy basically to like see if they can target the prion protein gene and induce the expression of like protective proteins. So that'd be kind of cool if they could do that. The stem cell therapy is also kind of, it's kind of cool too, but at the same time it's just like stem cells, if you know what stem cells do. They're basically trying to see if the stem cells, with their regenerative processes and properties, can help to repair and um, like regenerate damaged neurons and brain tissue. So that would be pretty cool if they could do that. Um, there's also immunotherapy that they're trying to see. This one is honestly kind of cool too, because if they could, if this is successful, it'd be really cool. Um, because they're trying to stimulate the immune system to clear prions, or they're trying to stimulate the immune system to basically like block the propagation of like misfolded proteins. So like they're trying to see if they can boost the immune system to a point where they can fight off prions, or they can stop or block like the beginning of like misfolding proteins, and I think that'd be super cool. Last one though, that's like the most promising and like the most progressed clinical trial treatment, all that kind of stuff, is charm. It's not called charm because it's in Lucky Charms. No, um, it's called charm because that is short for coupled histone tail for auto inhibition release of methyltransferase. And that's just a whole lot of mumble jumble to say that it is basically just DNA methylation. So they're trying to methylate DNA and modify it so that way it can safely suppress prion protein levels in the brain. And that has honestly been successful. They've been successful with doing it in mice, and they say that it is safe to do in humans, so it looks like they might be doing it soon. So, these are some upcoming and future treatments, and honestly, most of them look promising. So, be hopeful.
Prion diseases can affect both humans and animals. For example, in animals, prions cause bovine spongiform encephalopathy or mad cow disease, scrapie, and chronic wasting disease. Mad cow disease occurs in cattle, causing behavior changes, coordination loss, and eventually death. While humans consume infected beef, it can lead to variant Creutzfeldt-Jakob disease. However, it is the only prion disease shown to be transmitted between animals and humans. Scrapie affects sheep and goats and has been around for centuries. Its symptoms are intense itching, leading animals to scrape their skin raw, and neurological decline. Chronic wasting disease is found in deer, elk, and moose and spreads through contaminated environments. These could include water, food, or soil which have been mixed with feces, saliva, blood, or urine. Infected animals will drastically lose weight, show abnormal behavior, and die within months or years. In humans, the most common prion disease is Creutzfeldt-Jakob disease, or CJD. It can be developed sporadically through genetic inheritance or through contamination from medical procedures. It progresses rapidly and can cause dementia, muscle stiffness, uncoordinated movements, blurry vision or blindness, trouble speaking, and trouble swallowing. Currently, there is no cure, and most patients would come within a year. While prion diseases are rare, prevention is critical to protecting both animals and humans. One of these strategies is medical safeguards. Since prions are highly resistant to traditional sterilization, hospitals and laboratories use decontamination protocols, including the use of chemicals and autoclaving at high temperatures to prevent transmission. Another strategy is livestock monitoring. Governments and agricultural industries collaborate to monitor and prevent prion diseases in livestock. For example, this can include banning the use of animal byproducts in feed to help reduce the risk of spreading prion diseases like mad cow disease. As well, animals are tagged and monitored so that gene history, immunization, and food information is easily accessible. A final strategy is public health education. This helps to raise awareness about prion risks. It helps minimize exposure by advising against consuming high-risk animal parts such as brain and spinal tissue. The search for treatment against prion diseases is ongoing with progress in recent years. One approach is heterologous prion proteins. Here, scientists explored introducing prion proteins from one species to block the spread of misfolded prions in another. The study showed that these proteins could act as decoys, preventing further damage to the brain. While it is still in early stages, this method holds promise for future therapies. Another approach is targeting the unfolded protein response UPR. This study demonstrated that activating specific cellular pathways could reduce neurodegeneration in prion-infected mice. An oral drug is aimed to enhance this and overall help cells handle prion misfolding. This approach targets PERK, a receptor protein, to reduce the harmful overactivation of UPR while also preserving its ability to manage misfolded proteins. Overall, tests showed delayed disease progression, and improved survival.
Why should you care about prion diseases? You should care because they're dangerous. Prion diseases are fatal and incurable. At the moment, there are no cures for prion diseases, and treatment is primarily therapeutic. You should also care about them because they have a long incubation period. In CJD, the incubation period is between 1 and 30 years, so you may have the disease but not know it. Lastly, you should care because prion diseases can be zoonotically transmitted, so properly cooking your meats is important.